So it's my pleasure to welcome Nancy to visit our institute here. So Nancy is a professor in the Department of Brain and Cognitive Science at the Massachusetts Institute of Technology and an investigator at the McGovern Institute for Brain Research. She studies neural and cognitive mechanisms underlying human visual perception and cognition. So it's my pleasure to invite you here and welcome to give a talk at our institute. The stage is yours.
Thanks so much. Pleasure to be here. So I was told that this is a fairly broad audience, and so I'm going to start with maybe the first third to a half of the talk is kind of older work, setting the context, and then I'll get onto some more recent work towards the tail end. So great. So I'm going to start with some good news. And the good news is I think that our field of human cognitive neuroscience has made a lot of progress in the last 25 years or so. And one way to appreciate that is to step back and think about where we were just a few decades ago, where the state of knowledge of the functional organization of the human brain was pretty rudimentary. Much of it shown in this slide here. So from studies of patients with focal brain damage, we knew the very approximate location of brain regions engaged in things like understanding language, recognizing faces, and directing visual attention to these very kind of approximate regions of the brain. But that was more or less it. There were a few other little things, but these were the main things that were known at the time. And then functional MRI came along. And here is one depiction of where we are now. And the point of this diagram is that there are now dozens of little patches of the cortex for which we have a pretty good idea of what goes on in that patch of cortex. And each of these regions is present in approximately the same location in pretty much every normal person. And of course, this diagram is highly schematic. So let me step back for a moment and say a little bit more about what I mean by sticking a word label on a patch of the brain by showing you some actual data. So this is a picture of the bottom view of the brain in a recent member of my lab who was a real hero, getting scanned a lot. So we mapped out lots of different functions in his brain. And what you see here is an inflated cortical surface. So of course, cortex is all folded up in the brain to fit in there. And so to be able to see the native cortical surface, we mathematically unfold it to inflate the brain. And so what you see in gray, there's my pointer, the dark bits are the bits that used to be inside a sulcus and then got mathematically inflated. So you can see that surface. And we're looking up from underneath the brain with the back of the head, the occipital poles here and here. I hope everybody's oriented. Okay, so that's what's in gray. The stuff in color are the regions we have studied with functional MRI. So for example, that little blue patch there, that's the fusiform face area on the lateral side of the fusiform gyrus. So named because the functional MRI response in that region is substantially higher when people look at faces, that's the blue bar, than when they look at other body parts like faces and hands and knees, or scenes in green, or objects or words in these other colors here. So right next door to the fusiform face area is that little green patch there, that's the parahippocampal place area. So named because it responds very strongly when you look at images of places or scenes, pretty much any kind of place you could be in, indoor, outdoor, familiar, unfamiliar, doesn't matter. If it looks like a place you could be in, you get a strong response compared to all these other conditions. And even though this is only a centimeter or two away from the fusiform face area, the response to faces is 0. In this case, that means the same as staring at a tiny dot and not moving your eyes. We can't turn the visual system off, but we can get it as quiet as possible by giving people a blank screen with a fixation point, and that's our 0 here. Over in the left hemisphere, in a similar location, but on the left, we see the visual word form area, so named because it responds strongly to visually presented words and letter strings. Importantly, this region is not about understanding the meaning of words. It doesn't care if it's a real word or a string of consonants that has no meaning. But it responds strongly to all of those conditions compared to everything else, faces and scenes and objects and stuff like that. And this region is of particular interest because it's pretty much the only patch of cortex for which we know that the selectivity of that patch of cortex is trained up by that individual's experience. And we know that because you don't find this selectivity to visually presented words until after people learn to read, not before. And we also know this because it responds selectively to orthographies that that individual knows. So if you can read both English and Chinese, that region will respond to both English words and Chinese characters. And if you are like me, really lame and only know one language, it only responds to English orthography, not to Chinese or Arabic or Hebrew or anything else. Okay, those are aspects of high-level visual perception. And out on the lateral surface of the brain, we have some more good stuff. These little purple patches here respond very strongly when you look at images of bodies and body parts. That's the purple bar here compared to faces and scenes and objects and other things. What about other sensory modalities? Well, the orange patches here, these regions respond strongly and selectively when you hear the sounds of speech. Those are these two orange bars here compared to all these other kinds of auditory stimuli. And importantly, this region is kind of the, in a way, the auditory analog of the visual word form area. It's not about the meaning of language because it responds the same if you're listening to speech sounds in a language you know and a language that you don't know and don't understand. So it's really more interested in the difference between a ba and a pa than in the meaning of words. So all of these bits that I've talked about so far are specialized for different aspects of high-level perception, visual and auditory. So a classic question that's been in our field for hundreds of years is whether we also have brain specializations for aspects of higher level abstract cognition. And I think that's a really important question. And that's why I was really psyched when around a decade ago, Evelina Fedorenko joined my lab. And we decided to tackle this classic question of language regions in the brain. So these red parts in the left hemisphere along the temporal lobe and up here in the frontal lobe are this person's language regions, identified in this case with a simple contrast of reading sentences versus reading non-word strings, with a difficult task on the non-word strings so that actually difficulty goes against the selective response to sentences. And so many people had seen this going way back. And as I mentioned at the beginning, we've known for 200 years that Wernicke's area and Broca's area were involved in language. So there's no real news there. But by identifying exactly the right regions in each subject individually, we could get serious about asking whether those language regions are really specialized for language per se, or whether they're engaged in other aspects of language. So what Ev Fedorenko did was to identify these regions in each participant, and then measure their response in a whole suite of other kinds of cognitive tasks that people thought overlapped with language in the brain. And so the details don't matter. We tested lots of stuff: arithmetic, music, working memory, cognitive control, all of these things that people had argued seem to overlap with language in the brain. And by targeting exactly the language regions in each individual participant, we could ask whether those regions respond in these conditions. And what I've found is if you look here, this is basically Wernicke's area. Here's the response of that region with functional MRI when people read sentences versus non-word strings. That's how we identified it. I mean, this is held out data, but that's just replicating the criteria by which we define the region. But the news is, look at the response in all of these other tasks, basically a big bunch of nothing. Similarly, if you look in what's basically, Broca's area up here, you see a very similar pattern: strong and selective response, understanding sentences, and not much of any response in any of these other conditions. And so I love this result because to me, it's not just another selective blob on the brain, although it is that, and I am partial to selective blobs on the brain. I think this is a particularly interesting one because it addresses a question people have been wondering about for a very long time. And that is, I seem to keep losing my advances. There we go. It tells us that language and thought are not the same thing in the brain. The brain regions that respond when you understand language are pretty much non-overlapping with those that are engaged when you do all of these other aspects of cognition. And actually even stronger evidence for this dissociation between thought and language comes from the very beautiful work of Rosemary Varley in England, who has been for many years testing patients with global aphasia. So these are people who have the horrible misfortune of having massive left hemisphere strokes, which deprive them of pretty much all of their language abilities. It's not just that they can't speak, they can't understand language, they've pretty much lost all language. And that's a horrible tragedy for the person, but it's an opportunity for us to ask about the relationship between thought and language. And what Varley has shown is that pretty much every function that you might have thought would require language doesn't. And so these people with global aphasia can do arithmetic problems, they can solve logic problems, they can appreciate music, they can hold information in working memory, and they can think about what another person is thinking. And that's pretty fascinating, I think, in terms of the sharp dissociations between our signature language skills and a lot of the rest of human cognition. Okay, so I just mentioned the concept of thinking about other people's thoughts that has been worked on in greatest detail by my colleague, Rebecca Sachs, who showed that this little pink patch here, the right temporoparietal junction, is extremely selectively engaged in thinking about what other people are thinking. And although many people have studied these functions in this general region of the brain, the signature of Rebecca's work is to show the incredible specificity of this region. So it doesn't just respond when you think about other people in general, like what they look like. It doesn't even respond when you think about another person's bodily sensations, like thirst or hunger or pain. It's only engaged when you think about the content of their thoughts. And if that seems crazy, reflect on the fact that this is actually, in many ways, the essence of what it is to be a human being, is to spend a lot of your waking moments thinking about what other people are thinking. We're a highly social species, and what we care about in other people is not so much what they look like. Okay, we pay a little bit of attention, not that much. We don't really care that much. What we care is what's going on inside, what they're thinking about. And so perhaps it's a little less surprising that there's a whole special piece of machinery in the brain for doing just that. Okay, so I want to hasten to add that I strongly believe that these regions are actually very functionally specific, but I don't think that's the case for all of the cortex. In fact, there are vast swaths of cortex shown in white here that are sometimes called the multiple demand regions. That's John Duncan's name for these regions because they respond to lots of different kinds of cognitive demand. So for example, if we look at all the conditions that Ev Fedorenko tested in the language regions, they all had a difficult and an easy condition. The difficult condition in solid bars and the easy version of the same task in outline bars. And what you see is across this wide swath of different tasks, those regions respond to the difficult versus easy version of all of these different kinds of tasks. So these regions are like the opposite of everything else I talked about before. They're very, very domain general, and they'll engage in lots of different kinds of cognitive tasks. In many ways, I think these regions are much more computationally puzzling, like what goes on in there, like what is this, the brain CPU or something? And so I think that's a really fascinating puzzle to understand what goes on in those multiple demand regions. Okay, so that is my very quick whirlwind overview of some of my favorite parts of the cortex, leaving out lots and lots of good work and just focusing on regions that I've spent some time looking at myself and am particularly interested in. But even from this cursory overview, you can see there's actually been real progress. We've discovered a set of functionally distinct brain regions that are present in pretty much everyone. And that includes regions specialized for very abstract, uniquely human functions, like understanding language and like thinking about other people's thoughts. And I like this diagram because I think it's a kind of initial sketch of who we are as human beings. These are the basic pieces, or some of the basic pieces of the machinery that's running in our minds. And I think that's cool. But at the same time, this is just the absolute barest beginning. I keep losing my cursor. There we go. And I think really the way to look at this diagram is as a roadmap. for research that just opens up a whole vast space of questions. And so what I want to do in the rest of the talk is kind of touch briefly on some of those wide open questions that are probably already in your minds from even just this brief intro. So one of the first and most important ones is, what is the causal role of each of these regions in cognition? And, just to state the obvious, in science, we're not interested in what tends to happen with what other things. We're interested in what causes what. That is really what our scientific theories are all about. And very sadly for me, a functional MRI is just never going to answer that question. All it's going to do is tell us, if you think this kind of thought or look at these kinds of images, these brain regions will turn on. That's a causal connection between what you do and engagement of the brain, but it doesn't tell you that those regions are necessary for performing those functions. So we're just not going to answer this question with functional MRI. And that's why I was very excited when a few years ago, I got a phone call from a colleague telling me that this gentleman here was awaiting neurosurgery in Japan, neurosurgery for intractable epilepsy that did not respond well to drugs. And so to map out their surgical route, the neurosurgeons had placed electrodes all over the ventral surface of his brain, shown here. And for comparison, to see why these electrode locations were of particular interest to me, here's the bottom surface of my brain showing my face areas in red, place responsive regions in green, color responsive regions in purple. And those electrodes seem to go right over the territory of some pretty interesting stuff. And so I was very thrilled that this gentleman had agreed to allow us to show him some pictures and record responses from his brain. And so you can see some of those responses here. So looking especially at the strip that goes right along the fusiform gyrus, the responses of each of those little electrodes here is shown in a little graph. Like, for example, 173 is an electrode right there. Time is on the x-axis. This is, what is this? I think around 500 milliseconds. And so what you see is red is a response of that electrode when this person looked at faces. And all of these other colors hovering around 0 are the responses to all other kinds of images, pictures of bodies and objects and characters and so forth, all produce pretty much no response there. So if you were underwhelmed when I showed you those bar charts from functional MRI with the response to faces here and objects there, and you're thinking, okay, big deal, it's not that selective. I agree. And I think when we have better data from higher resolution measurements, we see really extreme selectivity like that right here. But my point about this is not so much the awesome selectivity, fun as that is, but the causal tests that are very occasionally available from clinical treatment of neurosurgery patients. So the clinicians had decided for clinical reasons to really test the causal role of these regions because they wanted to perform surgery going in that vicinity. And so they electrically stimulate these electrodes here. And I'm going to show you a video of what happens to this person when he's electrically stimulated there. So he's speaking Japanese, but there's subtitles. And remember, here he is looking at the neurosurgeon, and we're seeing the stimulation here. This guy is the perfect subject. Like, he wants to tell us exactly what's happening. And here he's getting stimulated. He doesn't know there's a face area in the brain. He doesn't know what part of his brain is getting stimulated. He's just asked to tell us what happened. So that shows us the causal role of that region in face perception. The face changes when he's stimulated there. What happens when he's looking at something that isn't a face and he's stimulated in the same location?
No change. 最初の1秒ぐらい。初めて1秒ぐらいがなんか、横になんか目、口って感じでちょっと一瞬見えたんですけど、何かなと思ってたら、気がついたらもうこの箱にしか見えないっていうか。なんすか、またさっきの横に、その目と口って感じで、ちょっと。eyes.
This is a kanji character on a card.
More, please. One more time. やっぱり顔に見えましたね。口らへんって感じ。
Okay, you get the idea. The point of this is it shows striking specificity, not just of the response of those regions, but their causal role in perception. When he's stimulated in the face responsive region, it doesn't change the percept of the box or the ball or the kanji. It just puts a face on top of it. And though these data are rare and precious, in another video you can ask me to see later, when he was stimulated right next door in these color preferring regions, he sees a rainbow on top of whatever he's looking at. Again, showing the strong causal specificity of these regions. Okay, that was all question one, causal role. Another really obvious question that pops to mind when you just look at this diagram is, okay, what else? What other things are in there? And I'll just mention a few of the ones we've been looking at in recent years. And the first one is, although many of these things are social, face responsive regions, speech responsive, language responsive, theory of mind, we are very social organisms, and many regions in the human brain are engaged in social cognition of dealing with individuals one-on-one. But we also look at groups of people. And so in one line of work, Leila Ishik and Cami Koldewin identified a region approximately right there, right nearby the theory of mind region, but not overlapping with it. And it seems to be engaged when you look at groups of people socially interacting with each other. So we showed videos of two people interacting versus doing independent activities. And it responds much more when people are interacting than doing independent things. And in fact, from that region, you can decode from the pattern of response across that region, whether that interaction is a competitive one or a cooperative one. One of the basic things that we humans care about in social interactions, and that actually infants are sensitive to within, I forget exactly, I think 6 months of birth, infants can detect the difference between a cooperative and a competitive interaction. Okay, so all of that is still parts of the, some of the many parts of the social brain. But in fact, we understand not just the social world, but the physical world. So if I show you a simple, boring picture like this, you don't just look at that and say, okay, orange juice, fork, table, duh. You pretty quickly see, okay, there's the glass is supported by the table. The glass could be picked up. But if you picked it up, the orange juice might spill. And if it did, it might stain the tablecloth, et cetera. You see all of these other properties of the physical world. And so in recent years, we've been trying to understand if there are particular brain regions engaged in intuitive physical reasoning. And this is all work in collaboration with my colleague, Josh Tenenbaum. The first functional MRI study was done by Jason Fisher here. And we identified a set of regions here that are preliminarily implicated in reasoning about the physical world. So for example, when you look at the very same stimuli, this is from work that Josh had been doing for a number of years using behavioral and computational methods. If you have to judge, for example, which side of if this tower fell, would more blocks land on the red side or the green side? That's the intuitive physics task. Versus, are there more yellow or blue blocks? a difficulty match task. You get greater activation in each of these regions. In another task, we showed that if you see these red and blue dots moving around in a way where they're kind of bouncing off each other and colliding like billiard balls with physical interactions, you get a much higher response in these regions than if the dots are moving around in a kind of social way, flirting, chasing, or evading each other. And in subsequent work, we've shown that these same regions hold abstract information about the mass of an object inferred from its motion trajectories and its collisions with other objects. They have information about the stability of a scene. So for example, you can decode the difference between a scene like that and a scene like that, where something is about to happen next here, but not here. And so there's loads of wide open questions. And I want to mention that these regions are not as hyper-specific for physics as, say, the face area is for faces. In fact, they're suspiciously nearby and overlapping with those multiple demand regions. So they probably do lots of different things. But our hypothesis is that these regions are engaged in understanding the physical world based on a kind of mental generative model of the physical world that can run forward simulations to determine what will happen next, what Josh likes to call the brain's physics engine. So this is ongoing work, but that's the hypothesis. Okay, so maybe somewhere in there, something about intuitive physics. But what I want to talk about next is a new-ish approach to asking what other specializations might exist in the brain. And in this case, instead of doing the kind of normal science thing of coming up with a hypothesis and designing an experiment and testing it in the brain, which is a very respectable thing to do, here we just collect a whole big pile of data and try to let the data speak for themselves by doing some math. And so this started with a line of work in collaboration with my then grad student, Sam Norman Hegner and Josh McDermott. And what we did was to scan subjects listening to 165 common everyday sounds, little 2-second clips of recognizable sounds, the most common sounds you hear. And we scanned subjects listening to these sounds and measured the response of each voxel in high-level auditory cortex, well, actually all of auditory cortex, to those sounds. And that gave us a big data matrix of 11,000 voxels across 10 subjects, and the response of each of those sounds, each of those voxels to each of 165 sounds. So we start with a data matrix like this, throw out all the labels, and just do some math that basically says, can we boil down this big data matrix into some simple components? And I'm skipping all the details. It's basically independent component analysis. And what came out of that was six components accounted for 80% of the replicable variance in this data matrix. 2 of them were kind of known components of auditory cortex, that is selectivity for high and low auditory frequencies. So we replicated tonotopic cortex. That was cool. 2 others were not exactly new, not exactly old. They were sort of mid-level acoustic-y properties. Another one was speech selectivity. I talked about that before. Here is this little green patch up in there. And that had been reported before, but we could see like really whopping selectivity for speech. But our 6th component was totally new. And here it is, showing you the magnitude of response of that component over the 165 sounds. And all of these blue and purple sounds that are producing a high response are music. And then you see that the response magnitude just kind of goes off a cliff for all the other sounds. And so this shows whopping music selectivity. And what's kind of astonishing to me about this is the incredible variability of the music sounds. It's everything from, a classical flute solo to a heavy metal band to an opera singer to a rap artist and a drum solo. And so these things are acoustically just wildly different, and yet they all produce a strong response in this component. Now, this component is mathematically inferred. It's not the actual response of individual voxels. It's mathematically inferred from the ICA analysis of these data that enable us to kind of demix the neural populations that cohabit voxels with functional MRI. So I thought that was cool, but I really prefer to see stuff in my raw data. And that's why I was thrilled when we ultimately were able to see the same thing with intracranial responses from individual electrodes. So here, for example, is an individual electrode in a neurosurgery patient showing you the, even more clear selectivity for music just in the raw high gamma response of that electrode, thus validating the method that we used to discover this music selectivity, this matrix factorization method, and also validating the actual finding of music selectivity in the brain. So I got pretty excited about these data-driven methods, and I thought, okay, let's go wild. Let's do this all over the place. And a particularly wonderful opportunity came along very recently when Kendrick Kay and his collaborators published a really magnificent data set and made it publicly available. This is the natural scenes data set. It's a bunch of subjects scanned at 7 Tesla, and each subject saw over 5,000 natural images. Usually I don't like to bother with other people's data because I think mine are better, but in this case, Kendrick Kay's data is better than anything I ever collected. They're beautiful, beautiful data, and I thought just wide open for this kind of data-driven analysis. So my postdoc, Meenakshi Khosla, decided to apply a similar analysis to the natural scenes database. And she used a slightly different analysis. She used Bayesian non-negative matrix factorization instead of ICA, but it's the same basic idea. Just take the big data matrix, no labels, no nothing, and try to account for the pattern, the data in that matrix in terms of a smallish number of components. So what Meenakshi found is that when you do that analysis, in her case, separately in each subject, and then look at the similarity across subjects, you find 5 components that are highly consistent across participants. And so here they are. I realize this is kind of a rat's nest here, but here are 4 of the components. And so let's look at component 1. They're rank ordered by how consistent they are across subjects. And I don't know if you can see this, but here are the top images in component 1, and they're all faces. And we had independent subjects rate how salient the face was in each of the natural images here, and that's color-coded here. And so you can see that the response profile of that component is highly correlated with the salience of the face in the image. And similarly, component 2, oh, did I say this backwards? I said faces because I can't, it's so tiny on my screen. Sorry, that was scene selective. It's component 2 that's face selective here. Component 4, the 4th most consistent one across subjects, was selective for text. You can't really see it in all these images, but if you blow them up, every one of them has text in them, all the top ranked images, and it's strongly correlated with salience of text in the image, and 5 is bodies. So on the one hand, like, duh, this is stuff we already knew. We're just replicating stuff we already knew about. But on the other hand, I really love this result. One, it's a strong positive control. When you use a new method and discover stuff you know to be true, then it's like, yes, this method really works. So I love that. But even more importantly, I think this does tell us something new. It tells us that selectivity in the cortex for scenes, faces, words, and bodies is they're not just weird little things we can find if we go looking for them. They are part of the dominant structure of the ventral pathway. Like, this analysis is as hypothesis neutral as we can get. The stimuli were not designed to validate these hypotheses. They were designed to just broadly sample, visual experience. And yet out pops 4 of the top 5 are things we already knew. And I just love that. But even better, there was a new one. You might have noticed all this white space in the middle of this slide. What is that new one? Well, here it is. I don't know if you can see these images well enough, but they are all food. And when we ask people to rate the salience of food in the images, we get this strong correlation with the magnitude of response of this component with food. And I will spare you the details, which we published this just a few weeks ago. And I'll just say, when we found this, we thought, yeah, right, sure, food. We have looked for that before and not seen it. And there are a million obvious low-level correlates of these things. There's round plates. There are more saturated colors. There's lots of red and green. There's lots of curvature. And we spent 6 months trying to make this result go away because we just didn't believe it. And actually, it's just true. It's not a confound of any of the low-level features as far as we can tell, with many, many different controls. It seems that we actually just have food selectivity in the ventral visual pathway that we didn't see before, because actually, if you look in individual voxels, you can see this, but it's very weak. Instead, if you use the component analysis, what it does, as well as being a data-driven discovery method, it demixes the responses of different neural populations that live within a voxel. Because it is basically what it does is accounts for the response of each voxel as a linear weighted sum of components. And so it can kind of factor them out and discover these strong selectivities that are stronger than the selectivities you ever see in individual voxels. Okay. So now, what other specializations? I've given you sort of a tour of some of the recent ones that we think we're finding, but let's get on to some different questions. This is fun and games, and we're still looking for new stuff, but there's some other really fundamental questions, like how does all of this stuff get wired up in development? It's so systematic across subjects. There's a lot of variation in the exact size and exact position of these regions, but there's just spectacular specificity, replicability across subjects. Like in every subject, the place regions are medial to then the color regions, then the face regions, et cetera. That's very, very systematic. And so how does that get wired up? Well, I'm not going to answer that, but I'll throw out a few tidbits. There's a lot of work going back at least 15 years arguing that in the ventral pathway, these regions develop very slowly, particularly the face selective regions develop slowly and are still developing into adolescence. And there's one very beautiful study from Marge Livingstone and Michael Arcaro that argues that experience is necessary for the face regions to develop. And that's based on scans they did finding face patches in monkeys, which have been reported using similar methods for a long time, for Marge and Doris Sau and Venrik Freivold and others. But then what they showed is if you raise baby monkeys without ever letting them see a face, you do not see face selective patches in those monkeys. And therefore they argued experience with faces is necessary for the development of those face selective regions. Okay, so it looks like the face area at least takes years to develop and requires visual experience. Really? That's interesting. Those data are compelling. But these are really important questions. And so I think it's important to go at them in multiple ways. And so the one problem with the human work is that almost all of the human work has been done in kids age 4 and up. And that's kind of too late. It's hard enough to scan 4-year-olds, but a lot has happened by age 4. And so scanning infants would be even cooler. On the other hand, it's nearly impossible. They squirm and wiggle. They don't stay in the scanner very long. Their parents get anxious. The whole thing is like basically a train wreck. But my amazing colleague, Rebecca Sachs, and her then grad student, Ben Dean, and her heroic best subject, her own son, Arthur, right here. The three of them collectively figured out how to scan infants. And they started getting some pretty nice functional MRI data from 6-month-old infants. Like this, these data here, where you can see that the preferential responses for faces in the hot colors and scenes in the blue colors bear some resemblance to what we see in adults, even in a 6-month-old. That's pretty awesome. But in that first work, there just was not enough data from each individual subject to really characterize how selective these regions were and whether they had adult-like selectivity. And that's when the amazing Heather Kosakowski entered the scene, a grad student working with me and Rebecca Sachs, who just got her PhD. And Heather has an ability to scan infants like I'm pretty sure no one else on the planet. It's amazing to behold. You can see Heather holding a screaming, squawking kid, and you're thinking, this is just ridiculous. This will never happen. And Heather does not give up. And sure enough, half an hour later, the kid calms down, goes in the scanner, and Heather gets amazing data. And having spent years doing that and scanning over 80 infants, amazing, Heather has found face, place, and body selective regions in infants by 6 months of age. And she's been able to show in independent held out data, adult-like selectivity of the face areas in these young infants. Selectivity of the place areas, here's scenes and the other conditions here, they go in the right direction, but there's a funny business with the baseline that we're still working on. The body selective regions are strongly body selective in infants. And so what all this shows is that actually these regions don't take many years to arise. They're present very early in development. But what about this question of visual experience? Even a few months after birth is a bunch of visual experience, much of it composed of an adult sticking their face right in front of the infant's visual field. So what about visual experience? So my postdoc Ratan Murthy and I decided to take another whack at this question in a different way. We scanned congenitally blind subjects while they touched 3D printed objects like these here. So these are about yay high, 3D printed objects of faces, places, hands, and chairs. And we presented these to participants in the scanner. This is looking into the scanner bore with a very high-tech device of a disc of cardboard, a 99-cent CD holder here, and a postdoc situated outside the scanner bore, rotating the disc every few seconds, so the subject could just sit there and feel each object as it went by. And then we asked whether you get face selective responses by touch in congenitally blind people. First, we did the control experiment of showing these stimuli, just these exact ones, visually to subjects who were sighted, and you see the face selective response as usual here in the fusiform gyrus. What do we see for the blind subjects? Is pretty similar. So not every subject showed a face selective response by touch in the fusiform gyrus, but over half did. And actually looking at all of the subjects in their most selective responses now in held out data, you see a similar selectivity of response in the blind subjects. I don't know if you can read the y-axis here. I've occasionally made it really non-salient, but actually the responses are substantially stronger in sighted subject looking at faces than blind subjects feeling faces, but the response profile is very similar. So no, you don't need visual experience to develop face selectivity in the fusiform gyrus. So, okay, that was all developmental origins, my humble snippets of offering that these regions are present very early. And it seems like at least the face regions don't require face experience. I'll just briefly mention the wide open question of how all of this structure arises over evolution. Mostly we don't know because the relevant ancestors are no longer with us. But we can look at our relatives, for example, macaques. And I find it very interesting and notable. This is work from Rosa Leifer-Souza and Bevel Conway with me a few years ago, where we showed on the ventral surface of humans this very systematic spatial arrangement of face selectivity in purple, color preference in blue, and scene selectivity in green, in that order: faces, color, places. And if you look in monkeys, the action is mostly out on the lateral surface of the macaque brain, but it's suspiciously in the very same order: faces, color, places. And it's hard to imagine if you were designing a cortex, like, oh yeah, of course, would stick color between faces and scenes. It's like, well, it seems totally arbitrary to me. And so the fact that this is apparently arbitrary arrangement is so similar between humans and macaques, to me suggests that probably a lot of high-level visual cortex is homologous between humans and monkeys, that is inherited from a common ancestor. In this case, that would be 30 million years ago. And I think that's very interesting. I think auditory cortex is a very different beast in humans and macaques. We use audition very differently from macaques, while we use vision very similarly. And I could say a little bit more about that, but I'll skip it for now and get to what I think is in some ways the most fundamental question, which is what is actually represented and computed in each of these regions. And that is an enormously important question that we need to grapple with if we're going to ever really understand what these regions are doing in any serious way. And as probably many, if not all in this audience know, there's been major progress in the last decade with the arrival of convolutional neural networks, And there's a bunch of things that are important about these convolutional neural networks. One is that they suddenly work really well on object recognition tasks. And that has given us, for the first time, a possible model of how visual recognition might work in the brain. Like, we didn't used to have any computational models that could do the task. And so we had no real hypotheses, no image computable hypotheses about what the ventral pathway might be doing. So that's big news already. Even more cool, work in multiple labs has shown that actually the internal responses of different layers of these networks actually show a remarkable correspondence to responses in primate brains, including humans, with responses of early layers of the networks corresponding to early stages of processing in the brain, mid layers to mid levels, and late layers to late levels, suggesting that there's actually some similarity between what these convolutional neural networks are doing and what brains are doing, despite the fact that the networks were simply optimized to do the task. They were not designed to model the brain. And so I think it's actually a kind of deep and cosmic fact that the networks optimized, with backprop and very different kinds of training data end up with a similar solution to brains optimized by evolution and learning. But so one of the things we can do then is ask whether we could use these convolutional neural networks as models of the regions I've been talking about, the face, place, and body regions. And so what Ratan Murthy did was apply a method that others had applied previously to individual neurons. He applied them to whole brain regions like the face, place, and body area. And at first, when he proposed this to me, it's like, what? There's like tens, hundreds of millions of neurons there. What does that even mean? But actually turned out to be quite interesting. So briefly, the method is you scan subjects looking at a whole bunch of different natural images, in his case, 185 images. You scan them on many, many, many sessions, so you have good retest reliability of the response of each voxel to each image. You then feed those same stimuli into your convolutional neural network and collect responses in the network to each of those images. You then fit a linear weighted sum model such that when you feed an image into the network, collect its activations, select a subset of those units with a weight on each, and sum them up, you fit the magnitude of response of, say, the fusiform face area to that image. And you fit that over a subset of the stimuli. And then the question is, does that work to predict the responses to held out stimuli? And the answer is, it works astonishingly well. We can predict, so on the x-axis is the predicted response from a CNN model built just like this. And on the y-axis is the observed response of the FFA, PPA, and EBA to that same image. Each dot is an image. And in each case, the dot is the predicted response is predicted from a model built leaving that image out. And so what you see is just, this is not a correlation normalized by the noise ceiling. This is a correlation of 0.9 between predicted and observed. Absolutely. So these models are incredibly accurate in predicting responses. Whoops, I am running out of time. So I will just say that that's cool, and it's not just a stunt. It's we can use it in various ways. I'll skip over some of this. We published this a year ago. We can use it to produce stronger tests of the selectivity of these regions, for example, by using GANs to generate the best image for the face, place, and body areas, which to my eyes, validate what our claims of the selectivity of these regions. But what I want to mention in closing is what I think is one of the coolest applications of convolutional neural networks to an even more fundamental question. And that is, why should the brain have specialized machinery, for example, for faces in the first place? Why is that a good way to design a brain? Why not just have general purpose smarts in brains and none of these nice little blobs that I've been talking about through this whole talk? And so for a long time, I and pretty much everybody who thought about it said, well, probably the reason we have face selective machinery is that face recognition just requires a different set of visual features than object recognition. And so it makes sense to allocate that task to different network units or neurons in the brain because they're just different features. But that was kind of loose talk. And actually, we can now test that very precisely with convolutional neural networks. So what Katerina Dobbs, then a postdoc in my lab, now with her own group at the University of Gießen, what she did was to train CNNs, in this case VGG, but she also did ResNet and AlexNet and the whole works. You either train that network on face recognition, or you train it on object discrimination using the ImageNet database with all the faces and animal categories removed. So once these networks are trained, this is a model of what an optimized face system should do, and a model of what a generic object recognition system should do. And then we can ask how well these networks do on held out faces and objects. And what you see is that the face trained network is much better at recognizing held out faces than objects, and vice versa for the object system. And to a machine learning person, this is like, duh, out of sample performance is worse, like we know that. But to me, it's a very useful way of kind of testing and validating this kind of hunch that the features useful for face recognition are just different than those useful for object recognition. But that doesn't yet tell us why the brain has to allocate these tasks to different units. And so I made the prediction that actually the really strong test is if we train one network on both face and object recognition. So just take the network and just throw in faces and objects and just concatenate those categories at the top end of the classification layer and train it interleaved on both. And don't tell the network, these are the faces and these are the objects, just mush them all together. That the networks would do badly at that, because they would be forced to compromise and find some feature space common to both faces and objects, and that would be suboptimal, and they wouldn't work as well, and that would explain why we have segregation in the brain. I was very excited about that hypothesis, and it didn't work. The network jointly trained on both tasks did just as well on each task as the singly trained network on the task it was trained on. So at first I was like, damn, I really like that hypothesis and I'm bummed that it's wrong. And then I thought, wait a minute, let's look inside that network. Let's ask, how does this jointly trained network do both tasks? And so what we decided to do was do lesion experiments on the network, basically do cognitive neuroscience on the network to discover its functional organization. And so we lesioned subsets of units at each layer here. And I'll leave out all the details because I'm out of time. But basically what we found is that jointly trained network had spontaneously segregated itself into a face processing pathway and an object processing pathway. And that's how it did both tasks as well as the singly trained network. And I think it's just like what we see in the brain. And I find that super exciting. It tells us that this is what an optimized solution to both face and object recognition entails. It entails allocating different parts of your system for each of those tasks. And I think that gives us an answer to a why question about the brain. So now we can go beyond just saying, oh, we have some of these and some of those and some of those, to saying, why is that a good way to design a brain? Of course, that raises a question of what other categories will spontaneously segregate in networks. And so when we were doing this work, it was back in over a year ago, when we had not found food selectivity in the cortex. And we had actually looked and not seen it. And so we had a strong prior that food is like faces in the sense that it matters evolutionarily. We look at it all the time in daily life, and yet we know it's not segregated in the brain. So we better not find spontaneous segregation for food and objects when we run the same kind of experiment, training it up on classifying different kinds of food and objects. So that was our strong hypothesis. And it turned out to be wrong. In fact, we found segregation for food and objects, much like we found for faces. This shows the degree of segregation as you go across layers. Another cool thing that kind of matches the brain. At early layers, you don't find segregation, like V1, V4 doesn't segregate food or faces, but at layer layers, you do. When we found this result, we're like, okay, damn, it's not fitting what we know about the brain. And then by total coincidence, Meenakshi Khosla started analyzing those natural zoom data that I talked about before, and found that actually this did make a correct prediction about the brain. And that raises a question I'd love to know, which is how much of the functional organization of the brain can we predict this way? Can we figure out how to train networks on really high level cognitive tasks like language and math and theory of mind and all this stuff and find spontaneous segregation for even the high level things. Okay, that's hard, but maybe we'll get there. But more generally, what I love about this work is this kind of work and other work, actually earlier stuff by Josh McDermott and Alex Kell had shown similar stuff with speech and music in networks. And I think this whole line of work is enabling us to go beyond just characterizing the functional organization of the brain to asking why it's organized the way it is. So I went over my time. I will just end with a summary slide here and say thank you for your attention, and I will take questions.
Thank you, Nancy, for the really nice talk.